Thank you, and thank you for the opportunity to join you here today. So I'm speaking on behalf of the H3 Africa Consortium, and particularly the H3 Africa Bioinformatics Network. And in fact, I'm standing in for Nicki, who's the PI of the Bioinformatics Network. She and I are both at the University of Cape Town in the computational biology group. So the H3 Africa Consortium, it's the Human Heredity and Health in Africa Consortium, is a program that's been co-funded by the NIH and the Wellcome Trust. That's been running. We've had our first 5 years funded, and we've just started the second phase from the Common Fund. And the H3 Africa vision has been to facilitate really an Africa-based contemporary research approach for genomics and environmental factors that drive common disease, and with the goal of improving the health of African populations specifically. So there's a big focus on two additional, apart from primary research, also looking at capacity development of infrastructure, both human capacity and the actual physical research infrastructure, and then also building collaborative networks within Africa. So it's really about doing African research by Africans in Africa for the benefit of African participants and populations. So it's a large consortium. These are all the different kinds of grants that have funded different programs. So we've got collaborative centers, 14. We have a strong LC component, that's ethics grants. Pilot biorepositories, we have 3 of those across the continent. And primary research projects, and then also global health training programs, also across the continent. And then the Bioinformatics Network sort of sits as an infrastructure within there, supporting all the different programs that are ongoing. So it really, the Bioinformatics Network has a very strong supporting role. This is just an overview of all the sort of the geographical spread of the different components of the consortium. So I'm not going to go into it in detail, but to say, yeah, overall about 25 research projects involving more than 500 investigators, 27 African countries involved. At the end of the first phase 1 of 5-year phase 1, we had approximately 75,000 participants recruited into the studies. And by the end of phase 2, we're aiming for 117,000 African participants. So phase 1 had an investment of over $76 million in the first phase. And as I say, we're now in the second year, heading into the second year of our funding. So I respect the text is probably very small, but this is just to give you an idea of the different types of common diseases that are being covered by different research project areas. And we've bundled them here by mental health, microbiome research, infectious diseases, which of course for us in Africa is still a large part of the disease burden, and cardiovascular diseases, which are also a very large NCD component, and all of these interplay, so the infectious diseases are never separate from the other NCDs. You know, there's a lot of crosstalk, and a lot of our populations are in epidemiological transition, so that also impacts how we undertake the research. And then other genetic diseases, hearing impairment, blindness, sickle cell obviously is a big one for us as well. So this is just to give you an idea of the kind of resources we're collecting. Phenotypic data, we get demographic information. Obviously, Africa is neither one country nor one population, and each country is also not one ethnic group. So we actually have very many African populations, so we can try and collect the demographics around that. Anthropometric data, and then obviously the disease and health related phenotype data. And then for the genetic variation, we have human and pathogen data being collected. And sequence data, some whole genome, some whole exome sequencing. And then I'll talk a little bit about our genotyping at the end. So we also genotyping using chip data. And we've got about 35,000 samples to be run on our custom chip that I'll describe at the end. And then we also have the microbiome sequence data, patient and sample phenotypes with the 16S and full genome for microbiome samples. And then obviously we're also looking at the human sequence that comes with the microbiome It's a kind of add-on. And then we have biospecimens, which are DNA at this point, which are being deposited in the biorepositories. So the bioinformatics network, in the first phase, it was a bioinformatics network. Sorry, I think that jumped. In the second phase, we call it the informatics network, and we're bringing in a component of health informatics as well to support the collection of clinical data and the data storage, etc. So the bioinformatics network is a component of the whole H3A consortium. We work very much on the capacity development side of it. So we've put in a lot of infrastructure, server capacity, compute capacity. We do a lot of training, and we've moved from a classroom. We started with a classroom-based model, which was a train-the-trainer model, and we now have a more online sort of mixed model that reaches a lot more young scientists in Africa. We're working on clinical informatics tools and data analysis tools. So the informatics network has 28 partners in 17 countries, with over 120 members. And we also facilitate the archiving of data, of the data generated by the whole consortium. We transfer, validate, store, and then submit the data to EGA within an appropriate framework that's been defined for the consortium. And then we also have, we're working on a data and biospecimen catalog. So this is kind of the metadata that will help people identify what we actually have as resources. So I just wanted to talk a little bit about the H3 Africa Consortium chip, because I think from phase one, this has really been a bit of a game changer for those of us doing genomic research from within Africa. So until this was developed, we really didn't have a genotyping chip that could give us the coverage we needed for African populations. They were very, as I think, I'm telling a story, everyone knows how poorly represented they have been. And particularly sub-Saharan Africa was underrepresented in many of the global consortia working on these things. So we used 3,500 African samples, and the map just shows you the distribution of where those samples came from, and generated whole genome data sets and used those to design the, we started with, I think, 2.5 million SNPs that would go on this chip that would really be informative for African populations. So obviously there was a whole process to building the chip, but we're aiming to do this genotyping on our Africa chip of over 36,000 H3A study participants, and have already done about just over 20,000 to date. So yeah, it's just to give you an overview of what's happening in the H3 Africa Consortium. I think that's all I've got there. So obviously our acknowledgements for the funding. So I have here acknowledgements funding for the bioinformatics network, but obviously for the whole program, there's also the Wellcome Trust and GSK funding that would be acknowledged. And this is just the informatics network team that I have named here, but obviously I can't put everyone from our whole consortium on the chip, although they're all very valid and valued members. Thank you.
That's such a fantastic effort, really. Any questions for Nicki? Rory, please.
Rory Collins, Oxford. Can you comment on how you're balancing the demands on open access to the data with the need to build local capacity and training and career development in Africa?
Sure. So the primary premise is that we really have African PIs and African participants and African researchers working on these projects. I mean, we have people do partner with external researchers, but the deal is that it's driven from within Africa by Africans. So that's the premise on which the collaborations happen. Then we have a whole defined infrastructure of moratoriums on the data that are generated and the specimens that allow the people who actually generated those data to have an amount of time to work on their own publications and to put those data to use in their own groups before those data become accessible to the rest of the world through EGA. We also have a very specifically constructed DBAC. It's a Data and Biospecimen Access Committee, and there's a whole process, obviously, for applying for access to the data. So we also try and look currently with the biospecimens, it follows a kind of Nagoya Protocol approach. So if people want to collect those biospecimens and use them for their own research, there's a strong component of partnering with Africans to do that research, and also any data that get generated through additional research must feed back in the same kind of structures. And I think that's also within a time frame, the biospecimen and collaborative agreements. So it's all spec'd out in a whole set of documents and, the SOPs and what have you.
Any other quick questions? If not,
Have you found that those have worked? I mean, it's all very well having the documents, but and has that given sufficient time for people to generate the papers? Has that worked as a way of driving?
So currently, the collection of samples in Africa is not trivial. So we have our first set of data, I think, under the moratorium in EGA already from the Awigen project. Audrey Duncanson can correct me if I'm off beam here, please. So we're stepping into that next phase. So none of our data yet are out there for access generally. So I hopefully can report back in 6 months or so and tell you how it's going. I mean, we have a continual debate about the consent structure and consent processes, and I think we all know there's a push for Nagoya to, broaden to include biodata. So I think that's a conversation that's going to be ongoing and dynamic. Yeah. Thank you.